This recording will go over part 1 of acid-base balance. It'll go over what a normal pH is, what's an acid, what's a base, what are buffers, what do they do, why is the carbonic anhydrase equation important, and what are some sources of acids and bicarbonate. The pH is an indirect measure of the free hydrogen ion level in the blood and other body fluids. Acid-base balance is the maintenance of arterial blood pH between 7.35 and 7.45 through the regulation of hydrogen ion production and elimination. pH is inversely related to the level of free hydrogen ion. And so when you see that it's inversely related, that means they're opposites. So in a solution, if it has a higher number of free hydrogen ions, the number of the pH value will actually be less. So if you remember, in general, a neutral pH is 7. If that solution, something happened, something was changed with it where it increased the number of hydrogen ions. So an increase in hydrogen ions would be a decrease in pH. So we would see the pH become more acidic. Same thing if we're sitting at a 7 and something happens that absorbs or takes away some of the hydrogen ions. A decrease in hydrogen ions would lead to an increase in pH. When one goes down, the other goes up. pH and hydrogen ions are inversely related. Again, balance homeostasis occurs by matching the rate of hydrogen ion production, which is happening all the time, with loss. And it's said best here that acid-base balance or pH balance is maintained through the regulation of hydrogen ion production and elimination. So as I previously stated, the normal pH for arterial blood is 7.35 to 7.45. That's a number that you're going to have to know. And arterial blood, when you're looking in the labs, you find it under the patient's ABG lab values. So ABG stands for arterial blood gases, and it's a blood draw that is done from the artery. So they pull out arterial blood, and that's what's analyzed. So the normal pH for ABG labs or for arterial blood is 7.35 to 7.45. And maintaining homeostasis, maintaining that blood in that normal 7.35 to 7.45 value is important because once it becomes off, it affects many things, including hormones, enzymes, how drugs work, the distribution of electrolytes, which affects fluid and electrolyte imbalances, and it also changes excitable membranes, making the heart, nerves, muscle, and GI tract either less or more active than normal. So what is an acid? An acid is a substance that releases hydrogen ions when dissolved in water or body fluids, increasing the amount of free hydrogen ions in that solution. So if we're adding acids to a patient's blood, so inside their body something's going on, they're getting more acids added to their blood, which is causing a release of hydrogen ions, increasing that amount of free hydrogen ions. You go back and you think about your pH scale. What does that mean for the pH if I have an increase of hydrogen ions? So my normal is my 7.35 to 7.45, like right around here. So when I have an increase of hydrogen ions, remember that moves my pH down. It would decrease and head down towards 6. Carbonic acid, H2CO3, is the most common acid in body fluid. A base binds free hydrogen ions in solution and lowers the amount of free hydrogen ions in the solution. So just the opposite of what we just said about acids, right? With bases, they lower the amount of free hydrogen ions, and we know that hydrogen ions are inversely related to pH. So if my hydrogen ions are lowering, that means my pH is going up, right? So it'd be a higher number going, depending on how out of whack we get, we can go higher than 7.45 and start becoming abnormal. Bicarbonate is the most common base in human body fluid. So what are buffers? When you think of a buffer, you should associate it with a sponge. So just like a sponge, a buffer can either soak up extra hydrogen ions to help keep that pH normal, or you could sort of squeeze the sponge. A buffer can release hydrogen ions if that's what's needed to keep a pH normal. So in a sense, they can act either as an acid releasing a hydrogen ion or act as a base binding a hydrogen ion. But the job of the buffer is to keep the pH between 7.35 and 7.45. So if you remember, bicarbonate ions are the most common base in the blood, and it's a weak base. So when you look at the ratio of carbonic acid, which is the most common acid in the blood, to bicarbonate ions, the most common base, this is a weak base, but you'll see there's 20 bicarbonate ions to every 1 molecule of carbonic acid. So although it's weak, it's very numerous. The relationship between carbon dioxide and hydrogen ions is something that you're going to have to get straight in your brain to understand all this. So carbon dioxide content of fluid is directly related to the amount of hydrogen ions in the fluid. And this equation right here is called the carbonic anhydrase equation. And it shows that the concentration of carbon dioxide is directly related to the concentration of hydrogen ions. So when carbon dioxide increases in the patient, so let's think about a patient who is experiencing respiratory depression. And it doesn't matter why. We'll say they've had too much narcotics. Okay, so they're experiencing respiratory depression. So they're not exhaling like they should be or how they normally would. So they're holding on to CO2 and that's increasing in their body. That leads to an increased concentration of hydrogen ions. These are directly related. Let's think of something else. Let's think about if there's an occurrence in the body where that causes an increase of hydrogen ions. And for this, we'll think about diabetic ketoacidosis, right? The name itself says acidosis. So we have an increase of hydrogen ions that leads to an increased production of CO2. Think about how the body compensates for DKA, diabetic ketoacidosis. Right? Think about how it affects the respiratory system. Right? You might see Kussmaul respirations in your patient who is experiencing DKA. And what that is, that deep, rapid respiratory rate is related to the patient trying to decrease the amount of hydrogen ions in their blood to decrease that acidosis. So the DKA has caused an increase in hydrogen ions. The body tries to compensate by blowing off carbon dioxide, which in a simplistic manner you could say is like blowing off acids. So they start experiencing Kussmaul respirations, deep rapid breathing, blowing off that carbon dioxide, trying to get rid of hydrogen ions. So again, this is your carbonic anhydrase equation. In the middle is your carbonic acid, which is not very stable. So it's going to flip-flop easily one way or the other. If the patient has a condition where that leads to an increase of hydrogen ions, it's going to cause a shift to the left, right? So an increase of hydrogen ions would cause the body to create more of this carbon dioxide. The patient can exhale that, and that will decrease the numbers of hydrogen ions in the blood. So an increase of hydrogen ions would mean that the solution or the blood is more acidic, right? And to get it back to being normal, there'd be a shift to the left where the patient would start blowing off more carbon dioxide, in essence, blowing off acid. It's my simplistic way of saying it, which leads to a decreased concentration of hydrogen ions in the blood. On the other hand, if the patient is experiencing a condition that causes them to hold on to CO2, such as respiratory depression, that will cause this equation to shift to the right. So I'm not blowing off CO2 like I should. It leads to an accumulation of hydrogen ions within the blood. So just some questions to get you thinking. How does respiratory rate affect pH? If you can keep down the DKA example, you can keep it straight in your brain, right? So with DKA, we know it's acidosis. There's extra hydrogen ions, and the body compensates by creating more CO2, trying to breathe off that carbon dioxide to lower the hydrogen ions. On the flip side, if there's a condition that's causing the patient to have decreased respirations, they're holding on to CO2, which would increase the hydrogen ion concentration in the blood. And since hydrogen ion is inversely related to pH, an increase in hydrogen ions results in a decrease in pH. So how does bicarb affect pH? So bicarb is our base. So an increase in bicarb, how would that affect pH? An increase in bicarbonate causes the amount of hydrogen ions to decrease and causes the pH to increase. Then on the other hand, a decrease of bicarb causes the free hydrogen ions levels to increase. Increasing free hydrogen ion levels would cause a decrease, and they're inversely related, a decrease to the pH, causing it to become more acidic. Acids and hydrogen ions are produced continuously through normal body physiologic work and metabolism. Normal metabolism of carbs, protein, and fat creates natural waste products. Carbs create carbon dioxide, protein, sulfuric acid, and fat breakdown creates fatty acids and keto acids. Incomplete breakdown of glucose, which occurs whenever cells metabolize under anaerobic conditions when there's no oxygen, forms lactic acid. Anaerobic conditions can occur with hypoxia, sepsis, and shock. Finally, cell destruction allows cell contents to be released, including the structures that contain acids. These release acids in the extracellular fluid increase free hydrogen ion levels. And this visual just again shows the relationship between carbonic acid and the weak but numerous bicarb. So 20 bicarbs equals, as in the body, to every 1 carbonic acid molecule. Bicarb is again a weak base, but it's very numerous. It's the main buffer of extracellular fluid. It comes from GI absorption of ingested bicarb, from pancreatic production of bicarb, movement of cellular bicarb into the extracellular fluid, kidney reabsorption of filtered bicarb, and the breakdown of carbonic acid.